Neuromyelitis optica is an autoimmune disorder where patients develop an antibody, a protein, that's usually used to fight off infections, but in the case of neuromyelitis optica, they make an antibody that targets their own optic nerve, spinal cord, and brain. So this antibody is doing something that the immune system never intended to do. So if it binds to the optic nerve, patients can have blurred vision or develop blindness. If it binds to the spinal cord, patients can have weakness, numbness, difficulty controlling their bowel or bladder, or even full-on paralysis, or become quadriplegic. At UT Southwestern, we have one of the largest biobanking efforts in neurosciences, where we've sought permission from patients to store their DNA, to store their blood samples for future research. So we undertook a genetic study in order to determine were there genetic risk factors for developing this disease. What was different about this study was that we used whole genome sequencing. How is that different than other studies? So imagine you had millions of lines of computer code and there was a problem in the code. You have two options. You can either read the entire code from start to finish, or you could take little samples and look at every third page or every fourth page to try and find the problem. All of the previous studies had done sampling. In our study, we looked at the whole code from start to finish, and that's what led to these novel, never seen before findings. It had never been done before in this condition, and in terms of rare diseases, this is one of the largest studies of whole genome sequencing in a rare disease. What this study has done is highlight the importance of a very specific biologic pathway in the pathology of this disease. There are already clinical trials going on of drugs that could target this pathway, but now we'll have a much better way of deciding which patients may or may not respond to certain drugs. Furthermore, what this study does is allow us to model the disease in a much better, much more accurate way to develop new therapeutic targets and to understand what we could do in the future.